Hello and welcome to the TLC of Amino Acids experiment. Thin layer chromatography was done in organic chemistry to separate organic compounds so we could see whether or not that organic compound or other organic compounds were present in our mixture. With the TLC of amino acids, we're essentially going to do the same thing, except we're going to use a cellulose plate, which looks very similar to a TLC plate, and we're going to analyze and try to determine an unknown amino acid. So for this experiment, one thing that's a little bit different is that the organic compounds we looked at were UV active. And so all we had to do is shine UV light underneath it, UV light on it, and the compound itself would absorb UV radiation. For amino acids, since very few of them have conjugated aromatic rings, the UV activity is not there for most of them. So what we need to do is modify that amino active and either make it UV active or make it visible just by the naked eye. So we are going to add an anhydride, which is this compound right here, to react with that amino acid and make it visible to the naked eye when we look at our TLC plate. So in one of the questions, I want you to just think about this mechanism a little bit and think about what it would take to get from an anhydride to this trione intermediate. So what we're doing is converting this diol, two alcohol functional groups connected to this carbon, into a ketone functional group. In the process, the layout is you need to do a proton transfer, and then a leaving group needs to leave, and then you need to do one more proton transfer. So one of the two alcohols needs to be protonated to make OH2+. The lone pair on the other oxygen will come down to form a double bond and kick off the leaving group in the second step. So that is leaving group leaves. And then once you have water in solution and you have a strong acid, so essentially a protonated ketone, you can use water to deprotonate that ketone to get you to your trione. Once we have our trione, our amino acid can react with the ketone and convert this from a ketone to an imine. So specifically, the bond that formed is between this nitrogen and the carbonyl carbon on that ketone. Several steps happened, and eventually what we did was did proton transfers to make this OH, and then we kicked it off with the lone pair on our nitrogen. So the nitrogen formed a bond with that carbon. We eventually made a double bond between this nitrogen and this carbon to get us to our imine. There was then a similar process between another equivalent of the trione and the primary amine that we formed to make our final product, which is what was purple in color on the TLC plate. Now notice that the only part of the amino acid within this compound here is simply just the nitrogen. So when we're looking at the colors on our TLC plate, what we're looking at is the nitrogen of the amino acid with other components of the anhydride dye. So in this case, we actually couldn't scrape off our amino acid from the TLC plate because we've converted it into a whole different compound where only the nitrogen of the original amino acid is present. All right, for the pre-lab questions, one of the important questions is draw the remaining amino acids. So I want you guys to look up the amino acids, and I'm actually going to pull one up here on the screen. In this case, for our amino acid table, the one that's not always seen is the selenocysteine. So here we have selenium instead of the sulfur to make our cysteine amino acid. For this experiment, we're using 6 amino acids. One of them is the lysine, where we have our carboxylic acid, an amine of our backbone, and then we also have an amine at the end of our carbon chain. We're going to use alanine, or that methyl group on alanine. Alanine is hydrophobic. We're also going to use isoleucine. So we have an isobutyl group as our carbon chain for isoleucine. And we are going to use phenylalanine, where we have an aromatic ring, tyrosine, a modification of that, where we have an alcohol functional group, and then finally glycine, which is a special case because we actually do not have an R group other than a hydrogen for glycine. So those are the amino acids that we will be comparing to and what could potentially be our unknown amino acids. All right, looking at this in a little bit more depth, there are several ways we could run this experiment. We could run it on a regular silica gel plate like we did within organic chemistry. One issue is that a lot of times we're not going to have much differentiation in RF values if we run it on a regular TLC plate. So that's one reason we might not do that. Also, based on the interaction potential between the plate and our amino acids, we might have to add some bases to stabilize our cysteine, phenylalanine, and tyrosine. And I'd rather not do that for preparation of the compounds, because if we had to prep these, then you would essentially know what your unknown was before you even ran it. Another option is a cellulose TLC plate, which is what we're actually going to do in lab. This gives a little bit better separation between all the compounds. One or two clear disadvantages is that you can't see some of them very clearly. So imagine you're running an amino acid ladder where you've got 6 different amino acids. If it was amino acid 2, 3, and 4, you'd have a very hard time differentiating those 3 as different when you run your ladder to compare it to the individual compounds. Now 2 has an advantage where it's a different color, so if these 3 were on here, you could probably at least say you had 2 and then either 3 or 4. But considering the number of amino acids that overlap at about the same RF value, so you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 amino acids that all have RF values very close to 0.3. If you go up a little bit, you could probably find another 4 or 5, and if you go for a little bit more, you could probably find another 4 or 5. So that is definitely a problem we're going to run into with this experiment. Now, fortunately, we were given this table, so we have an idea of the specific RF value that we expect for these different compounds. Since we are using the cellulose TLC plate, I highly recommend that you use this data to predict the polarity and therefore the distance traveled on the TLC plate for each of your compounds. So I've actually combined this into a Word document, and I'll post this and make it available for you as an experiment. But I have the amino acids as well as the expected RF values for the cellulose plate. So the expected RF values for the cellulose plate is always in brackets here. So I've actually ordered these where we expect the RF value of lysine to be 0.24, the lowest RF value. Lysine, like we said, has a protonated amine at the end. This is the most polar. And then, like we said, glycine is a special case that only has hydrogens there. It doesn't have an R group. This is actually also pretty polar. If you don't have an R group there, it's going to have more impact on the polarity of the actual compound than it did before if you have something like an isobutyl group, which is a very non-polar group. Another interesting observation is that alanine and tyrosine have very similar values. So tyrosine has the alcohol functional group on the aromatic ring. That definitely adds some polarity to it. But like we talked about with glycine, since there really is no R group, it makes the polarity of the carboxylate and the amine a little bit more prominent. Here we only have a methyl group, so it doesn't take away a lot. It doesn't give a large nonpolar region to decrease the polarity of this significantly. So we expect these to be very close in area to each other, but the tyrosine, based on the values given on this site, is a little bit less polar than our alanine. And then one final thing to note is this is not the best representation of these amino acids. So when you're asked to draw the amino acids, do not draw these structures. And the reason why is because in this experiment, we're running the plate under acidic conditions. So we have acetic acid, and then we're spotting these in hydrochloric acid when we run the experiment. So that being said, since we're under acidic conditions, the carboxylic acid will remain protonated, but all the amines will be protonated as well. So all of these NH2s actually need to be NH3 with a positive charge. So when you're drawing the structures, make sure you specify that we're under acidic conditions when we run the experiment. So make sure you protonate all of your amines and give them a positive charge. All right, welcome to the TLC of Amino Acids lab. Your TLC plates are in this box here. Take out a TLC plate. Let's examine this. So we have our gel on this side, and this is the plastic side holding that gel. It's a little bit more shiny. So I'm going to lay that down with the plastic down because we're going to end up spotting on this side of the plate. For what we're running up the plate, we're going to have our 6 amino acid standard mixture. So we've got 6 amino acids there, and we're going to assess unknown G and unknown AA. And then we also have a standard amino acid mixture with 5 amino acids here. We'll discuss in more detail what's in each of those standards. So first thing we need to do is weigh out 1 milligram of our unknowns. So we're going to go over to the scale and weigh out 1 milligram of each unknown. All right, we're going to start with G, and we're going to get 1 milligram of G. Let's first start by looking at this. So this is actually in units of grams. We could adjust this or just realize that 1 milligram is that unit right there. So if this reads 0010, that would be exactly 1 milligram. As long as we're pretty close on these, that's all that matters. We can also adjust the volume that we add if we're a little bit over in terms of the mass that we add to these test tubes. So we'll see where we're at now. So that looks very close to 1 milligram, so we might be at we're at 1.4 milligrams right now, so instead of 1 milliliter, I will add 1.4 milliliters or 1.3 milliliters to make our solution that we can then spot on the plate. All right, next we're going to do unknown AA to make our solution that we can spot on the plate. So based on what we saw last time, it doesn't take very much to get 1 milligram. I guess that's a little bit more. All right, we've got 2.4 milligrams, so in this case we'll add 2.4 milliliters of our hydrochloric acid solution to dilute this a little bit. All right, so we're going to dilute our samples with hydrochloric acid solution. So like we said, for G we had 1.4, so I'm going to add 1.4 milliliters of hydrochloric acid solution to G. All right, so we're going to dilute down G. Obviously we can dilute that down to the exact recommendation for A, since we have 2.4 milligrams in there. This is actually going to be over concentrated, but that's okay. We'll just spot a little bit less on the plate. So I'm going to run this down to 400, add that to both of them. So 400 in G and 400 in A, and we just need to be aware that A is two times as concentrated. And then we also need to dilute our ladders, so our 6 amino acid ladder and our 5 amino acid ladder. And now we can go ahead and pull 400 microliters from each. So first from our 6. Now our 5 amino acid ladder.
Alright, and now we can cap these. And lightly mix each one. And before we spot them on the plate, we'll make sure we stir those up a little bit. So now for the TLC plate, we're going to go ahead and mark this. White pencil line there, and then we want 4 lanes on this. So we're going to mark it there, 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 and there to run up the plate. So hopefully on the video you can barely see that. I can see it in person, which is all that's necessary because we don't want to scrape too much silica gel off of the plate. Alright, so we've got our ladder here, which has our standard with our 6 known amino acids. I'm going to stir this up a little bit here. And then go ahead and spot a small amount on the plate. My first mark. It's a little bit different than what you've seen in the organic lab because this is an aqueous solution. So it's hard to get a small spot. I think that works. But you really have to press it on the plate and then it all just wants to come out of the plate, or out of the spotter. Alright, so once I get the rest of this out, I'm going to go ahead and... Alright, so we're going to rinse this pipette out thoroughly each time. So basically let the hydrochloric acid solution run up it, and then spot it, let it run out, and then after we've done that about 5 times, which I did on the video, we can go to our next. So this is unknown G. Spot that on the plate, and then again wash our spotter several times. I didn't let G run quite as far up, so about 4 should be sufficient to wash this spotter out. And then AA was basically twice as concentrated as recommended. So with this, I'm going to make sure that I only have a very small amount that can run up in here. And when I spot it, hopefully this spot looks quite a bit smaller than what I did for the other one. It's pretty good. So we'll rinse this a few times. And then go ahead and spot our 5 amino acid reference ladder. Alright, so for setting these up, we're using these large glass plates. These plates essentially make it so that the solvent that's running up the plate doesn't evaporate as it runs up the plate. To hold these plates up, we've got this. And our solvent is going to be in this chamber here. And this is textured on the inside, so we want to make sure that that inside that is textured is pressing against the plate. That way the solvent doesn't run up against the flat glass faster than it runs up the plate. So we can make sure our 2 textured sides are facing in. So we've got our 2 textured sides facing in. We can now put our plate right here. We spotted everything very high, so we didn't need to worry about having our spots dissolve in the solvent, which is convenient. So once we have that, then we can clip both sides. Of our plate. And then next we need to make our 9:1 ethanol to glacial acetic acid solution. So that's 45 milliliters. And then we're going to add 5 milliliters of glacial acetic acid. I'm going to go ahead and pour this in here. We may need to add a little bit more. We'll see how much that is once the solvent starts running up the plate. And so it looks like our solvent is running up the plate. There's actually a line on the glass here, so it's hard to see where the actual solvent level is. You got to kind of tap it. So it looks like our solvent level is actually above that center line. And it looks like we're maybe a ½ centimeter above the plate. So let's go ahead and add a little bit more. Let's go ahead and add about 20 milliliters. I'm going to go ahead and make another solution, add a little bit more to make sure it keeps running up the plate, and then we'll take another look once it's run about halfway up the plate. Alright, so we've run about halfway up the plate. It took about 1.5 hours, almost 2 hours, but we made it halfway up the plate. So we're going to go ahead and take this out of here. Drip some of this excess solvent in here. And as we check out the plate, I'm going to go ahead and mark where the solvent line ran up to, so that we can assess RF values of our amino acids. Before we heat this plate, we need to actually spray it with ninhydrin so that our amino acids can react and actually be visible as we look at it. Lightly spray this plate. Now we'll set it on this warm plate and watch it dry.
On the plate, we've got this on a hot plate, and the solvent is drying off of it, so the water and ethanol are certainly evaporating off of the plate, but also our ninhydrin is slowly starting to react with those amino acids. And remember, that was a multi-step process where all we are actually taking from the amino acid is the nitrogen, but there are several steps to actually get it to the point where we have a visible compound, which is the ninhydrin amino acid derivative. So as the plate continues to warm up, you can actually see, if you look very closely, kind of in the shadow in the upper part of the plate, some spots to form. That's the center of the plate, so it's the hottest region. and you can actually start to see some of the amino acids. And those 2 amino acids that you see are actually our 2 unknowns. So based on the fold of the plate, it looks like the center is touching the plate a little bit more than the outside, so that's why that area is heating up quicker, allowing that reaction to happen quicker. Now on the left and the right, specifically in the top part, you can start to see the ladder form. It looks like it's also pretty hot down toward the bottom right-hand corner of the plate, which is where that 5 amino acid molecular weight ladder is. So essentially our reference ladder. And we're going to lift it up here just to give a slightly closer view. So again, you can see the 2 unknowns and you can see the molecular weight ladder. I think we can see almost all that we're probably going to get from our molecular weight ladder, as well as our 2 unknowns. And I think based on the way those line up with the molecular weight ladder, we should be able to at least figure out what our 2 unknowns are. Now that we have the plate and we have reacted our amino acids with anhydride, we can take a look at the RF values of each of the compounds and determine what our 2 unknowns are. So looking at the plate, we've got our reference ladder where we have our 6 amino acids over here, we've got our 5 here, we've got unknown G right here, and we've got unknown AA right here. So starting from top to bottom, clearly our 2 unknowns look like they've traveled pretty far up the plate. Those are the least polar amino acids that we have. And specifically, the least polar amino acid we have for sure is isoleucine. The 5th most polar or 2nd least polar is our phenylalanine. Our phenylalanine. And if you need to look these up again, we looked at the structures of those earlier in the video, so feel free to go back to that if you need a little bit of review. But we have our isoleucine drawn here, which is the least polar, and our lysine drawn here, which is the most polar. Now, I do want to go actually basically talk about all of them in order just because we are missing an amino acid from our 5 ladder. So our 5 amino acid ladder, we're missing one. The second we talked about for sure was glycine, and then the third actually was tyrosine. So tyrosine, so this is the order from least polar to most polar. And remember, even though that tyrosine has the alcohol functional group, we're under acidic conditions, so it's not protonated. It's not like we have a cation there. And we have that large aromatic ring, so all those carbons in the aromatic polar area. So that's our order in terms of polarity. For our solvent front line, it looks like we're between about 11.9 and 12.1 centimeters. If you want to go with millimeters, that works too. So between 119 and 121 millimeters. So let's call it 120 millimeters for our solvent front. So 120 millimeters for our solvent front. And then looking at our individual amino acids on here, this is not easy to interpret. So I'm going to actually just start out and essentially put a circle over each of them. It was actually pretty convenient. We had these 2 unknowns to give us an idea of what our top 2 are in our molecular weight ladder. So this first unknown definitely had the highest RF value. So if we measure from our baseline to where this compound traveled, it looks like that is about 10.75 centimeters. So we would take 10.75 divided by 12 to give us the RF value of our least polar compound, which is isoleucine, which looks like it matches up pretty well with this spot here, which unknown G then clearly you can come to a conclusion of what that's going to be. For the next highest compound that traveled, our 5th most polar compound or our 2nd least polar compound, we have phenylalanine, and that one also pretty clearly matches up with our other unknown. So hopefully you can come to a conclusion of what unknown AA is. Now, the reason why I wanted to talk about this in more detail is because we need to make some conclusions on where our last amino acids are. So we decided that the fourth most polar is alanine, so that spot right there matches up with this spot right here. This could be debated that it's 2 spots, but from what we looked at with the RF values, we know that glycine and lysine are going to be very close to each other. So glycine is here, and lysine is right underneath that. It looks like the drag didn't show up very well for where we had our 6 amino acids. I'm not sure why that happened. It looks like solvent splashed on the plate, but from what I saw, that definitely didn't happen from the way that we ran it. It's possible that it dragged a little bit more in that area on the glass. Maybe the glass indents didn't push the plate far enough to allow decent separation of those, but we actually have 3 amino acids bunched up in this area where we had 2 in this area. So the one that we're missing is right here, which is essentially this area right here. So a lot of the dark purple there, and then a little bit of this dragged slightly higher, is the other amino acid. And that other amino acid, hopefully you can conclude, is tyrosine. So from our 5 amino acids, we have lysine, glycine. We do not have tyrosine there. We only had 5 in our ladder. And then we have alanine, phenylalanine, and isoleucine. So from our 6, the 2 that are underneath this, again, are lysine and glycine. And then we have tyrosine, which is here and not here. And then the top 3 match up, which is alanine, phenylalanine, and isoleucine. So we were able to use our ladder of our known amino acids to then match those values up with our unknown amino acids and figure out what those are.